Repel.
ADP-ribose, polymerase 1 inhibitors. Hello everyone. Welcome back to our channel. Today, we're exploring the intriguing realm of molecular biology, with a particular emphasis on a crucial enzyme called PARP1, or ADP-ribose, polymerase 1. This enzyme has garnered significant attention in drug discovery for almost 30 years, and there's a compelling rationale behind it. The insights we're sharing today are drawn from the Journal of Medicinal Chemistry, 2010, Volume 53, Issue 12, authored by Dana V. Ferraris from the Johns Hopkins University Brain Science Institute. So, let's begin. First off, what exactly is PARP1? Often referred to as the guardian angel of DNA, PARP1 plays a crucial role in maintaining our genetic integrity. It's an abundant enzyme found in the nucleus of our cells, and it's been the focus of over 20 medicinal chemistry programs targeting various health issues, including cancer, stroke, and diabetes. Now, despite the extensive research, it's only recently that PARP1 inhibitors have started making waves in clinical trials, especially for cancer therapy. This is exciting because PARP1 inhibitors show promise in two major therapeutic areas, ischemia and cancer. So, why is PARP1 such an attractive target for drug development? Well, it all comes down to its enzymatic function. PARP1 is a 113 kilodalton protein that has three major structural domains. The first is a DNA binding domain, which contains two zinc fingers. These fingers help PARP1 latch onto damaged DNA. The second is a catalytic domain that uses a molecule called nicotinamide adenine dinucleotide, or NAD+, as a substrate. This is where the magic happens. When DNA is damaged, whether from oxidative stress, radiation, or even certain chemicals, PARP1 jumps into action. It binds to the damaged DNA and gets stimulated to produce long chains of ADP-ribose. These chains are like little flags that signal other repair proteins to come and fix the damage. Now, let's talk about the process itself. When PARP1 senses DNA damage, it binds to the site and its activity can increase up to 500 times. It starts adding ADP-ribose units to histones and itself in a process called poly-ADP-ribosylation. This modification loosens the grip of histones on the DNA, making the damaged area more accessible for repair enzymes. But here's the kicker: as PARP1 does its job, it also consumes a lot of NAD+. This rapid consumption is crucial because it means that the cell is actively trying to repair itself. Once the repair is done, another enzyme, called poly-ADP-ribose glycohydrolase, or PARG, comes in to break down those ADP-ribose chains, allowing PARP1 to detach from the DNA and get ready for the next round of damage. In summary, PARP1 is not just a guardian angel, it's a dynamic and essential player in the DNA repair process. Its ability to sense and respond to DNA damage makes it a key target for therapies, especially in cancer treatment, where it can help overcome resistance to DNA-damaging drugs. So, what exactly are PARP1 inhibitors, and why are they important? First off, let's talk about PARP1. Poly-ADP-ribose, polymerase 1, or PARP1, is an enzyme found in the nucleus of our cells. It plays a crucial role in detecting and repairing DNA damage. Think of it as a guardian angel for our DNA. When DNA gets damaged, PARP1 rushes in to help fix it by adding chains of ADP-ribose to proteins, a process known as ADP-ribosylation. Now, here's where PARP1 inhibitors come into play. These inhibitors block the action of PARP1, which can be particularly useful in cancer treatment. Why? Because many cancer cells have defective DNA repair mechanisms. By inhibiting PARP1, we can push these cells into a state of synthetic lethality. This means that the cancer cells can't survive without their DNA repair capabilities, leading to their death. PARP1 inhibitors have been a hot topic in medicinal chemistry for nearly 3 decades. Interest surged in the late 1980s, and by the 1990s, researchers were exploring their potential in various therapeutic areas, including cancer, stroke, and inflammation. Fast forward to 2005, when groundbreaking studies revealed that BRCA1 and BRCA2 deficient cancer cells are particularly sensitive to PARP1 inhibitors. This opened up new avenues for targeted cancer therapies. Today, several pharmaceutical companies are actively developing PARP1 inhibitors. Let's take a closer look at some of the key programs. 1. Bipar/Sanofi, their lead candidate, BSA-201, is currently in phase 3 trials, focusing on triple-negative breast cancer, an aggressive form of the disease. 2. Kudos/AstraZeneca, they have developed KU-59436, also known as olaparib, which is in phase 3 for metastatic breast and advanced ovarian cancer. 3. Abbott and Pfizer, both companies are working on their own PARP1 inhibitors, targeting metastatic breast cancer and melanoma. For Initech and Merck, these companies are exploring PARP1 inhibitors for various solid tumors, showcasing the versatility of this approach. 5. Mitsubishi, they are investigating PARP1 inhibitors for cerebral ischemia, which could potentially expand the therapeutic applications beyond cancer. How do these inhibitors work? Most of them are designed to compete with NAD+, the molecule that PARP1 uses to perform its function. By binding to the active site of PARP1, these inhibitors prevent it from repairing DNA, leading to an accumulation of DNA damage in cancer cells. As we look to the future, the potential for PARP1 inhibitors seems bright. With ongoing clinical trials and a better understanding of genetic factors in cancer, these drugs could become a standard part of treatment for patients with specific tumor types. The concept of BRCA-ness, referring to tumors that exhibit similar vulnerabilities to BRCA1/2 mutations, could expand the use of PARP1 inhibitors even further. PARP1 inhibitors represent a cutting-edge approach in cancer therapy, targeting the very mechanisms that allow cancer cells to survive. Next, let's explore their current clinical progress and what the future holds for these innovative therapies. First, let's talk about what PARP1 is. ADP-ribose, polymerase 1, or PARP1, is an enzyme that plays a crucial role in repairing DNA damage. Think of it as a guardian angel for our cells. When DNA gets damaged, PARP1 springs into action, helping to fix those breaks. However, in cancer, this repair mechanism can sometimes work against us. Now, PARP1 inhibitors are designed to block this enzyme, leading to the accumulation of DNA damage in cancer cells, especially those with existing repair deficiencies, like BRCA1 and BRCA2 mutations. This concept is known as synthetic lethality, where the combination of two defects, one in the cancer cell's DNA repair and the other from the PARP1 inhibition, results in cell death. Currently, there are several PARP1 inhibitors in various stages of clinical trials. 6 groups have already moved their inhibitors into human trials, with 2 more on the brink of phase I studies. Notably, companies like Bipar and AstraZeneca have advanced their candidates into phase 3 trials, targeting cancers like metastatic breast and ovarian cancer. That's pretty exciting, right? And it doesn't stop there. Abbott and Pfizer are also in late phase 2 trials with their own PARP1 inhibitors. This is a clear indication that the pharmaceutical industry is placing significant bets on these drugs as viable cancer treatments. What's particularly promising is the discovery that PARP1 inhibitors can be effective not just for BRCA mutation carriers but also for other tumors exhibiting a BRCA-ness phenotype, tumors that have similar DNA repair deficiencies. This expands the potential patient population significantly. Now, let's talk about ischemia. The Mitsubishi Group is leading the charge with a PARP1 inhibitor specifically targeting cerebral ischemia. If they succeed, it could pave the way for other PARP1 inhibitors to be used in treating ischemic conditions. Looking ahead, the future of PARP1 inhibitors seems bright. As our understanding of cancer genetics deepens, these inhibitors may become standard treatments for various cancers with identified DNA repair deficiencies. The goal is to tailor therapies to individual genetic profiles, much like we've seen with drugs like imatinib for specific cancer mutations. In the ongoing battle against cancer, researchers have been exploring innovative compounds that can enhance the efficacy of existing treatments. One such compound, known as Compound 77, has shown remarkable promise in preclinical studies. When tested in models of melanoma and breast cancer, it demonstrated significant tumor growth inhibition when used in combination with established chemotherapeutic agents like temozolomide and carboplatin. In a groundbreaking move, Abbott Laboratories initiated a phase 0 human clinical trial for Compound 77, which allowed them to administer low, non-toxic doses to a small group of patients. This early-stage trial aimed to gather crucial biochemical and pharmacokinetic data to inform future studies. The results were encouraging, revealing that the compound was well tolerated and effectively inhibited PRP levels in both tumor biopsies and blood samples. Building on this success, Abbott has embarked on several phase 1 trials to evaluate the safety of Compound 77 in various cancer types, including leukemia and solid tumors. Notably, they are also exploring its potential in treating brain cancer, with trials designed to assess its effectiveness in combination with radiation therapy and in pediatric patients with recurring tumors. As the research landscape evolves, other companies have also made strides in the development of PARP inhibitors. For instance, Initech and Genentech have been investigating their own candidate, ANO-1001, which has shown promise in enhancing the effects of chemotherapy in patients with advanced melanoma. Despite facing challenges in clinical trials, the compound has demonstrated the ability to prolong life and delay tumor growth in preclinical models. Meanwhile, Guilford Pharmaceuticals has focused on developing inhibitors specifically for neurodegenerative diseases and ischemic injuries. Their lead compound has shown potential as a chemopotentiator in brain cancer, significantly increasing the lifespan of subjects in preclinical studies when combined with temozolomide. The journey of these compounds reflects a broader trend in cancer research, where the integration of novel therapies with existing treatments could lead to more effective strategies for combating various cancer types. As these studies progress, the hope is that they will pave the way for new, targeted therapies that can improve patient outcomes and redefine the future of cancer treatment. In summary, PARP1 inhibitors are making significant strides in clinical development, with promising applications in both cancer and ischemia. As research continues, we can expect more personalized treatment options that could revolutionize how we approach these diseases. Thanks for watching. If you found this video informative, don't forget to like, subscribe, and hit that notification bell for more updates on the latest in medical research. Let us know your thoughts on PARP1 inhibitors in the comments below. Until next time, stay curious.
PharmaPal.